because of travel issues, she could not be here in person. So Emilie is going to join us remote. So Emilie Millet is our first keynote speaker, and I will turn it over to Emilie.
Good morning.
Good morning.
Perfect, it's working. So good morning, everyone. Truly sorry not to be here in person. Well, it's still a mess in Schiphol, so they canceled the flight and it was too late. Other flights were canceled because there were also some strike in France. The best combination, I think, to travel for a conference. But well, at least I'm connected, so I will try to show you, maybe present you some example in the data analysis and management in the European community. And after that, I'm sure you will see other example of data analysis for this last but not least session, you were right. A little bit of context, but I think we all know that by now, but with the climate change, we will see a negative impact on the crop yield. We already see it actually. This is a simple illustration on maize yield over the world with only 1 degree global increase, well, which is already a lot. There will be a lot of negative impact on yield. In combination to that, not all varieties respond the same way to this change of environment. This is the genotype by environment interaction, which is major form of variation. It's an opportunity to develop new varieties adapted to specific conditions, but it's also a challenge because it's complex biological mechanisms. It affects multiple traits with a response to yield. So we need to dissect this interaction to understand the gene to phenotype relationships. And I'm quite sure it's very clear for you by now after these days that all the progress in phenomics and modeling makes that possible. We can dissect the D by E because we now have access to a multitude of physiological traits measured in phenotyping platform. And just to be sure in my talk today, what I mean by phenomics is all this type of platform that we can classify in high precision platforms with very small level of measurement, the cell or the organs at minute or a day, but also the high throughput platforms. They can be controlled, it can be greenhouse or field, in which we can measure the plant or the canopy at a time step between the minutes to the weeks, but also the field multi-environment network. This is also phenotyping. It's not high throughput, but it's still phenotyping, and that can be very pretty precise, in which we measure canopies at the time step between weeks to months. All the traits that we measure in this platform can be relevant for yield prediction or to understand the plant's response to environment, but with more or less direct access to the yield. Also, something that is quite clear for everyone, but not always for new users, is that there are a number of challenges linked with these platforms. First, because there's a lot of variability of these platforms. That's the first thing. And they have constraints and limitations. One thing is that they can be more heterogeneous than fields. That's not always clear. This is one example to illustrate this point. In a greenhouse platform in Montpellier, there's variability of light during the day, different between the days, but also spatial variability. So this is the heat map from the top with each position on the platform. This is also the case when you grow plants in the growth chamber, where you can also have this light heterogeneity due to the light, of course, the lamp, but you can also have a different heterogeneity in temperature that can also change if you apply different water regime. Another source of diversity is the traits that we measure, and we can have plenty of traits, plenty of shape of a dynamical trait. For example, this is the photosystem efficiency in the former platform in Wageningen. Quite sure it's still accessible in the new NPEC. The leaf area over time in another platform, or the tip of the root growth. So there's different level of measurements, and a large number of data points that are generated. So it means that with this high dimensional data, how do we manage such large data sets? And also, how do we analyze them? So we need to adapt existing methods or develop new one. And we need also experimental designs for this platform because there's variability. We cannot just grow all the plants wherever we want. The example in the European community, I will show you things that we developed in the IPPN2020 project. So a few words on this project that is already finished. Actually, this was the European Plant Phenotyping Network, a research infrastructure project with the idea to help the community to progress towards the European infrastructure emphasis. We provided access to a number of phenotyping installations, but you can see some of them here at different locations in Europe. But there were also 3 joint research activities with the idea to help the community to also progress in the science and the tools and the technology. So the first joint research activity was about the technologies and methods. The second one was developing the tools for statistical analysis, and the last one was about information systems. So today, I will talk about a little bit about information system and about the statistical analysis. We'll show you some example of the tools and what we can do with those great phenomic data. First, why using information systems? And sounds very trivial now, but still, I think we have to make it clear that we need to organize and store the data sets so that they can be reanalyzed. This is what we need to do, reanalyzed and shared. Because students and postdoc leave, usually they leave with the hard drive. They rarely organize the data before they leave. Also because the hard drives and the USB got lost, stolen. So best using an information system rather than that. I'm not blaming anyone. I'm part of the problem. I was part of the problem initially too. So we can all improve. Also because it's clear now that we need several phenotypic experiments and we need to analyze them jointly because phenotypes will vary with environmental conditions. So to understand them, we need different environmental conditions. So we need different experiments. They cannot be reproduced. One experiment will have so many specific environmental conditions that it will be unique. And they are expensive. We're using new tools, we're using pictures, we're using a lot of human power to make it work. And one experiment cannot be reanalyzed if we don't use metadata and if they are not tracked. Very simple example. If you use the plant number, this will mean nothing for another experiment, for another platform, if you use different species in the same platform. So all the data need to be fair. They need to be findable, accessible, interoperable, and reusable. So you need information system for that. At the level of the European community in the IPPN project, to enable the data reuse, we defined the 3 levels. The first one is the internal traceability of the data. So at the platform level, at your platform, you need to have reproducible name of variables, data formats, and metadata collected. The second level is to reach fair data, not yet the interoperable level, but all the others. So there's a need to have machine-readable names of variables so that they are reusable, to identify all objects, and also to have, at least at the platform level, an information system. And the last level is to reach complete fairness of the data, is to use an information system that is connected via web services. So you could share the data easily. So I will show you some example of each of these. First, about the metadata and the unique identifiers of all objects in the platform using URIs, Uniform Resource Identifiers. So this is an example in 2 platforms. One is a greenhouse where you can have a different angle of the plant measure. So you will have all these URIs only for one event, one image. So the URI of a plant that should be fixed all along the experiment, the URI of the pot that the same for this spot in this platform, the URI of the cart, of the cabin, of the camera that you use with the specific setting of the camera, and of the picture. If you move to the field where you can also take some pictures, it will be the same. You will have the URI of the plot, because in the field, then it's a plot of several plants that you measure. The URI of one plant, if you're interested in only measuring things on one plant, of the leaf, if you track the leaf over time, again of the camera and of the picture. It's a lot of work at the platform level, get all of these URI, but for some of them, when it's done, it's only stored in the information system and it's there. And it helps also going back to what happened during the experiment. If a plant breaks during the experiment, then you can track back all the events linked with these plants in the experiment. An information system, an example that was developed in the European community is PHIS. There are others that were used in the EPPN, like PIPA. I just showed one pictures before. But for PHIS, there's of course a level of data management to store, organize, and manage the data. They can be any kind of data coming from the phenotyping experiments, can be pictures, can be spectra, can be environmental data. All the sources of data that we actually use for the analysis. They can be multispatial and temporal. We can have all the dynamic measured and comes from multi-source. So it can combine platform in the greenhouse, in the growth chamber, and in the field. But on top of that, the information system helps to enrich the metadata. It's not only storing the data, it's also adding the knowledge and other metadata to everything that it will be useful to reuse the data. And finally, it also enables interoperability because it can also integrate data from other resources, but it can also be integrated to other resources. If you have modeling platform, you can then connect your information system to the modeling platform or to another database. So that's the big picture for the information system. So at the level, at the analysis now, if I move to the analysis, once you have all the data collected and the information system set and running, we also need to have a similar work done for the data analysis. So we also define levels to help the community to progress for the analysis of this data. And we define two levels. The first one is to describe installations in the statistical intelligible way. What do we mean by that? Is trying to have a dialogue between the platform users, or the platform managers, and the statisticians. Because usually how platforms are described when we start is that we have nice pictures, we have maybe the map of the platform. It can be provided by the builders, but provided also by the platform managers. And we need to translate this into something simpler and to which we can then apply some statistical reasoning. So for example, moving to this kind of representation where we can see all the lane of the pots to a grid, a simple grid with row and column coordinates. And this is the starting point of all the experiment from the statistical point of view. In a second level, partners of the project were asked to describe the properties of the platform. What we mean by that is there is a lot of knowledge in this platform that are useful for the analysis, for designing the experiment, but also to analyze them. This is the example in the platform in Louvain-la-Neuve, where they grow plants in aeroponic tanks. So this is the level of information they have. So they know already, they suspect that there could be some gradients of temperature around the tank, and inside each tank, they suspect also a gradient of water availability. So this information is not only a metadata, but is also important for designing the experiments and analyze them. We also propose new strategies to analyze those phenomic data. Compared to classical field trials with multiple environment and one-time measurements, where we can first correct for the spatial variability and combine environments, or do one and two at a time, in phenomics experiments, We can have multiple experimental series, so different run-off experiments in one platform, but also multiple time measurements, also multiple scales. So how do we model the spatial and the temporal trends in the platform when we have high dimensional data? Well, we propose different modeling steps from using the experimental design, then extracting features from the pictures that were taken or from the spectra, correcting for design and factors from these new parameters, model the temporal trend, then combine that with other sources of variable, such as the environment, and the last step could be predicted target traits using this Phenomic data. So from a large number of basic and raw secondary phenotype data, secondary compared to the target traits, we end up with few parameters that are more genotype specific or treatment specific, but they have a high predictive power or high understanding power, let's say. So today I won't talk about the feature extraction, things that you can extract from pictures. I'm sure you've seen a lot of talk already, and you will see some also this morning. I will talk about what you should do first before running the experiment, which is choosing the appropriate experimental design, and some example of the other steps of the data analysis, and then what can we do with all these new data, the power of the Phenomic data in a way. So talking about experimental design, quickly, I won't go into much detail on the statistical design, because that could take actually the whole keynote per se. But to save you some time, I will give you the highlights for experimental design. The main goal is to randomly allocate the treatments, all the treatment, the genotype, if you have water treatment, if you apply nitrogen or other treatments. So they need to be allocated randomly in the platform. But that's not only that when you think about experimental design. You need to define what is your unit. What are you going to measure? Is it a plant? Is it a leaf? Is it something else? So you need to think about that also beforehand. I mentioned the environmental variability. Do you already know in the platform what will be the direction of the environmental variability? So then you can define some blocks, set of experimental units that are more internally homogeneous. This is illustrated here with the heat map of light in the platform. If we have to divide the platform into blocks, there will be those three. So there is a gradient in this direction for light. So we can see that all the experimental units in this right block will be more homogeneous than these other two blocks internally. And also you can restrict randomization using the grids that I show you, the row and column coordinates to assign the treatment. So avoiding having one variety, one genotype being always in the same row in this direction. So you restrict so that it's spread a bit more evenly in the platform, and this is called Latinization. So there's already two simple things that you can do to have a better allocation of your treatment in the platform to take into account this variability. So we proposed a simple procedure to help choosing the design. First is translating the map of the platform into a simple grid and investigate the disturbances in the platform. That's level 1 and 2. Then describe your experiments, how many rows and columns are you going to use, how many experimental units, how many treatments do you use, and then choose the appropriate software, check the design, visualize it, and extract the experimental map. So that's also one metadata. Once you then run your experiment, extract what I call raw data. In that case, raw data is something that can be extracted from the pictures or from another sensors. So it's not exactly raw data in the sense where it's not a pixel anymore. It's not a picture anymore. Sorry, it can be pixels, green pixels, or something else that is relevant. Then we propose a two-stage approach to analyze this type of data. In the first stage, there's going to be environmental correction, and in the second stage, temporal evolution of the genetic signal, so dynamical modeling. In the first stage, the idea is to accurately separate the genetic effect, the treatment effects, from the spatial effects, and do that at each time point. So the idea is illustrated here. For one time point, there's the raw data. This is the heat map of the leaf area at one time point. And we want to separate this raw data into the spatial effect, the genotypic effect, the effect of the variety, and the error, because, of course, we're interested in modeling the genotypic effect or the treatment effect only. And we will repeat this modeling at each time point measured in the platform. So we're going to use the following model in which we can have factor of or covariates that are of interest. If we use a specific water scenario, temperature scenario, and genotypic effect. And we will have factors or covariates that we want to remove. We don't want to include the replicate effect, but we have to model them. We don't want to include the block effect, but we have to model them and in a way remove them from the genetic signal. Similar story for the spatial effects. We want to model them, but we want to ignore them from the phenotypic effects. So applying this correction allow us to keep the data at the experimental unit level. So we don't need to predict at the variety level, but we can keep the information at the plant level if we measure something at the plant level. So we keep more power for further analysis for integration with other sources of data. An example of this first step is the leaf area over time. So in gray, I hope you can see some of the gray curve is the raw data, and the blue is the spatially corrected data. And usually applying this first correction, you see a kind of shrinkage because you had variability between plants that was due to the position of the plant. And we already reduced this variability by removing this kind of error in a way. In the second step, we want to model the time series. And for that, we use nested P-splines. The idea is to use three-level nested hierarchical growth model, P-splines. Why three levels? Well, that's somewhat classical now in phenomics and phenotyping experiment that we use panel or different genotypes, different varieties. So the first level is the plant level. Well, if you use leaf, it's the leaf level. All these plants or all these leaves assemble into the other level, that is the genotypic level, and all the genotype groups of genotypes can be combined into what we call population. So a classical example is shown here on the wheat data coming from the ETH, where all the genotypes can be grouped into region of origin. So this is varieties coming from a different region, and we can see that once we look at the curve at the population level, well, we have different response, so different height over time for different groups. So at each level, we model the time course using those P-splines in the mixed model framework. Using P-splines is actually good because there's it's flexible modeling. So if you remember, I presented that we now have access to different shape of curve, while P-spline can handle these different shapes. Also, because we have access to first and second order derivatives, we can extract parameters that are of interest, such as maximum speed or rates of growth. In speed, and we can check for these parameters if they vary between the different regions, so between the different groups. So these are those switch rates, but we can also measure the maximum of the plant height directly, or the area under the curve, which is an indicator of the overall performance of the plant. And already we can see that there's different variability for this trait between the population, and also different correlation. And this is only for 1 year of experiments. So applying these two steps, we can already have relevant parameters, and we can then combine them, because if you run different years of experiment for the same panel, then you can look how the traits change between the year of experiment. And then if you have the environment measured in all these years, then you can look at the response of these traits to the different environmental condition between the years. On top of that, something that is also a large question in Phenomics and phenotyping platform is the outliers. So using the splines also enable detecting some outlying time series. This is the only example I will show about outliers. But what we call an outlying time series is when we check the consistency of the time course of all the plants in a genotype. So this is an example here. If you look at all the time courses of the photosystem efficiency for one genotype of Arabidopsis, one curve is one plant. We're going to check if all the plants of the same genotype are consistent. So to do that, we look at one genotype in one treatment, it could be one management, and we compare the coefficient from the p-splines of all the replicates. We are looking at the metadata, those coefficients. We try to look if they correlate, and we try to look if the slope of the correlation is also close to 1, because we don't want to deviate from this correlation. And here, for example, we can clearly see that there's one plant that does not correlate with all the others. So it's a signal that something might be wrong with this plant. We also look at the PCA of the coefficients, and we can see that one of the plants also deviates from all the others. So user can define thresholds for the correlation, but also for the angle between the plants. And these plants will then be tagged, and as a result, you will receive a table of plants that are strange. So a metadata of outlying time series. So it's high dimensional data. We need to streamline the analysis. We don't want to do that everything by hand, especially now that we have high throughput platform. We don't want to draw the design by hand randomly. We want to use dice to randomly allocate the plants. We also don't want to use not automated analysis. So we need pipelines for analysis. So in this European project, colleagues from VSNI developed a new tool to generate the experimental design. At the moment, it's still called the design generator, at least it says what it does. So the input of this web application is user knowledge and the platform app. So this first level defined in the platform. And as an output, you get an experimental map, which is the first metadata of your experiment, actually, this experimental map, the very first one. And from the analysis perspective, what is the input? Is the raw data. So what I call raw data is the data after picture extraction. And the output is cleaned and analyzed data, or it is the corrected data. You also get a list of outliers that you can then input in the information system and what models were used. So all of these are also metadata that are very important for the reuse and for data integration. So in the European projects, we developed an R package called Stats and HTP for high throughput phenotyping platform. Data analysis that was also developed with other colleagues. And this package contains all the steps I've shown for correcting the data and modeling the time course, and also different outliers identification. And I will now use the remaining time to show you, I call that where the fun begins. It's not fair because it's a kind of fun also to do the rest of the analysis, but it's, let's say, at the level where you have all the clean data, all those nice traits that are measured in the phenotypic platform, and you try to combine them to understand the response of plant to the environment, or you try to understand what's happening with a panel of varieties that you're looking at. So today I wanted to present you the genetic progress in maize in Europe. So I think it's a nice study that with colleagues we managed to handle. So for that we use a panel of 4 successful European hybrids from the 50 to 2014. All these hybrids were grown in a network of field experiments with 24 experiments. And traits were measured also in 10 detailed experiments in different phenotyping platforms and 3 equipped fields. So this is the network of field experiments, some equipped field where we could also have pictures taken, but also high throughput flat platform. You've seen already some of them. The main result, I think the first and big result of this study is that the slope of the genetic progress for this panel of variety is about 100 kilogram per hectare per year. So there's a lot of genetic progress in this variety. So breeders worked well. This is the year of release of the variety, and the grain yield also grouped per scenario. So when we used the environmental information, we could group the different experiments into temperature scenario, but also soil water scenario, well water and water deficit. And what we observed is that the progress is the same inside each of those environmental scenarios. So it seems that genetic progress was good, but there's no difference between the scenarios. So we can ask what was selected by the breeders when they actually selected for grain yield. So they only looked at the grain yield, but what other traits were actually affected by this selection. So we combine tens of traits. I didn't dare to put hundreds, but I think we might be close to hundreds of traits measured in all the experiments with the environmental characterization and different modeling. So it's only one line that I'm writing here, but actually this involves all the steps, everything I presented so far. So this involves information system, data management, and also all the different modeling steps to reach the level of clean data that we can integrate between all these platforms. So when we look at all the other traits that were available, what we observe is that the genetic progress was mostly linked to phenology and architecture. So architecture, in a way, more efficient vertical distribution of leaf area. And from the phenology, there's a longer vegetative period and a shorter interval between the male and the female flowering of maize. So I present you this as something very simple, but actually all of those traits come from different sources. So I will show you just 2 examples. Results of a more efficient vertical distribution of leaf area, comes from 3D reconstruction and high throughput platform measurement. So this is how we colleagues obtain these results. So this simple answer already come from different modeling. And from the phenology point of view, this comes from combining data measured in all the field experiments and the temperature and the weather data. So already trying to answer the question, what was selected, and looking at tens of traits comes from massive work from different colleagues. So there's also a need for interoperability of the data if you want to combine properly what was measured on all these hybrids in different sources. So that's the first result. This progress is mostly linked to phenology and architecture. But what we also observe is our maybe favorite trait, what is physiological adaptive processes measured in the phenotyping platform, were not affected. But this presents a large genetic variability. They also have large heritability. So I will only illustrate with one trait, that is the stomatal conductance available in the platform. So we observe that for these traits, there were no genetic progress. So nothing changed between the old and the new hybrids, despite a range of variability in the genetic progress, and also an even larger range of variability when we look at diversity panel. So there's variability, but it was not, in a way, selected when selected for grain yield. So this also simple traits come from different step of modeling, multiple experimental series. Again, to have this simple result, simple with quotes, there's a combination of different data. So we have some hypotheses for why those physiological adaptive processes were not affected, and that calls for integrated more data. First, we look at the genetic correlation between the yield and those traits inside the different environmental scenarios. And what we observe is that those constitutive traits that were selected, like the flowering, the phenology, or the architecture, they were all positively correlated with the yield in all environmental scenarios. And those other traits, such as the stomatal conductance, but also light interception or the water use efficiency, are not always positively correlated with the yield. They are not always correlated with the yield at all. What we observe is that there's G by E or QTL by environments in the field. So breeder can only select for what consistently appear year after year. They cannot select for what disappear between the years. And this is what we really observed when we went one step further in the integration. So when we combine that with genomic information and we look at the QTL. So all those stable constitutive QTLs, so we have the example with 3 QTLs here, the heat map of allelic effects averaged in each scenario. So the allelic effects in the cool and well water scenario, we observe that there's a positive effect in all all environmental scenarios. So we improved yield in all experiments. And there's a good genetic correlation with the yield in all experiments. And also we observe an increased proportion of these alleles in the recent hybrids. So they were clearly selected and they have stable effects over the experiment. So it's another way of looking at allelic effects between the experiments. Now, when we look at some QTLs of the other traits like stomatal conductance or transpiration, more adaptive processes, then we see that the allelic effects will be different between the experiments. So there will be clear effects in some conditions, in other not, and they can change. For example, this one has negative or no effects in well-watered and positive effects in water deficits. And what we see is that the proportion of this allele not increase, it sometimes decreases. And also these allelic effects are unstable between experiments, so they cannot be selected directly. So it means that there's a reservoir of favorable allele for breeding new variety. So this is a simple conclusion. This is a nice example of what we can do when we integrate even further data. So all of this comes from Phenomic, but integrate with other levels, such as environmental data and Phenomic data. A simple summary of this is that Phenomic platform can help us understanding the plant responses to environment if we combine ecophysiological, statistical, and genetical modeling. I think that this is a great time for us. I think it's actually exciting for us now to be at the moment where we have already a lot of data available, but we can generate more data at different levels to understand what's happening for the plants. I think it's really a good time for the plant science now. But there are still challenges, and I think we are also trying to address those challenges now because we need to integrate the data at different scale and across different platforms. So we need a proper data management, but I think now that information systems are available, at least in the European community, I'm quite sure it's the same for other communities, and it's really necessary. It's a prerequisite if you want to use reuse the data. On the other side, there's the data management, but the data analysis, there's also a need for using appropriate tools and methods to handle and to analyze such large and diverse data sets, also if you want to use them in an automatic fashion. So there's a need for pipeline of analysis, not only for pictures analysis, but also for what comes after the pictures, what you've extracted. And for that, there's new tools and methods available in all communities. But I think this also calls for a wider perspective, because I presented also different disciplines, different modeling, but I think we can also use these other disciplines. There are things coming from ecology. We can also use mathematical modelings to try to make the most of this data and trying to understand the plant response. And with that, I would like to thank first the organizers of the conference. Apologize again, I could not be there in person with you. I acknowledge all the colleagues that made this presentation possible and all the partners of the IPPN 2020 project. And I would like to thank you for your attention. And I'm ready for some questions.
Thank you, Bill, very much. You were the cherry on the cake, I would say.
There's something I did not hear anything.
Oh, you can't hear me? Well, if you can't hear me, there is nothing we can do. Which is good for everybody. Can you hear me?
Yes, I can.
Okay. Well, number one, thank you very much for your talk. It was very holistic, integrative, and I should say you were the cherry on the cake, which everybody likes. I do have a question, but also that comes as a comment. Because I don't think you ever mentioned heritability, but was pervasive across the talk because of the things you addressed, which goes back to what I call also the boring QTL. I'm a QTL guy, and breeders can go after QTLs that are fairly stable across environments, but they can try to go after the more dynamic QTL. Clearly, it's better to go for both, in a way. But unfortunately, climate change is making things more and more difficult, so I'm more inclined toward perhaps boring QTLs that you can predict more easily. I wanted to know if you had any comments on this perhaps stupid question. Thank you.
I'm not sure it's a stupid comment. There's no stupid comment. Just to be sure, who was talking, if I may ask?
What is it?
Who is asking the question? I'm sorry.
Ah, Roberto Tuberosa.
I wasn't sure it was you, Roberto.
I was the one that converted François Tardieu to QTL a long time ago.
Perfect. That was a good idea to do that. First, I'm not sure it's boring QTL because those QTL that change between environments are extremely useful if we can model them. So that's the thing. Now we have more precise environmental conditions in each field, so we can actually make something with those QTLs. So I hope that's a little bit answering your comment or commenting your
Yes and no, because again, there are, yes, because the more unpredictable the environment it is, the more difficult is also for the modeler to tell to the farmer what to use, because for example, the largest drought 2 or 3 years ago was in Germany, not in Italy. So that's why I go for boring QTL. Because I'm pretty sure, so that's the point, pretty stupid, and also because I want to clone QTL, and I've done that. And so it's much easier to clone a boring QTL as compared to a more dynamic QTL. As long as they are both heritable, it's okay.
Yes, so you're right, and actually that's what was done for a long time, and that was successful. That's the story of the genetic progress. Looking only, selecting only using those boring QTLs was very efficient in terms of progress for yield. But I think we're reaching limits now by using only these. I think we will need to look at other traits that are more responsive, but also we can also think of boring QTLs at another level. Because now breeders, I'm not a breeder myself, so I don't want to talk for them, but what was clear is that they can define what we call target population of environments. So instead of selecting for the whole world or a big region, they can define, well, in a sense, those environmental scenarios and select inside those environmental scenarios. So what will be a boring QTL in one environmental scenario is actually this QTL that we observe being unstable across scenarios. So a boring QTL over the world It's not the same as a boring QTL in one scenario. They're not. I don't like calling them boring. I'm sorry, Roberto. I will stop saying boring.
No, I respectfully disagree with you, but respectfully, because they are much easier to work with and to clone. And some of them, actually, they're the most important QTLs that the breeder has to deal with. Phenology, of course, but this is trivial, what I call. And of course, you can use covariate analysis to get rid of the effect of this QTL. And indirectly, perhaps, the breeders have selected for this. And if you look at the historical series, they'll tell you a lot of work. So, again, to each his own. I like to work with both, but I much prefer to work with boring QTL. That's all. Thank you very much, by the way. Thank you.